Welcome back, class. Today we're going to be taking a look at plant pathogenic bacteria and phytoplasmas. And while as a group the bacteria are not as important as fungi and viruses in causing plant disease, some bacteria do cause very serious diseases. For example, Ralstonia solanacearum, formerly known as Pseudomonas, almost destroyed the banana industry in Central America. This particular bacterial pathogen causes MoCo disease. Another example involves the bacterium Erwinia amylovera, which causes fire blight disease, and this is the most destructive pathogen of apples and pears worldwide. You might remember one important bacterial pathogen called Agrobacterium tumefaciens. We talked about this in living systems too, and Agrobacterium tumefaciens causes a disease called crown gall disease, but it's become very important in the genetic engineering of plants. And the reason for this is that it can transfer DNA into plant genomes. Most bacteria can be placed into one of two broad categories, either gram-positive or gram-negative. Most plant pathogenic bacteria are classified as gram-negative, and they're readily visible at times 400 and times 1000 under your regular compound light microscope. When infected plant tissue is squashed or macerated in a droplet of water on a microscope slide, then covered with a cover slip and examined at times 400 under the microscope, masses of bacterial cells can be seen oozing from the tissue, and this is not seen with fungal infections. Then pure cultures of the bacteria can be obtained by picking up some of the bacterial ooze using a sterile loop, otherwise known as an inoculating loop, and then streaking that over the surface of nutrient agar in a petri dish. The bacterium then in pure culture can be identified based on metabolic tests. Gram stain, its ability to convert certain sugars, whether or not it contains certain enzymes, and also its resistance to different antibiotics. In this photo, top right of screen, we can see bacterial cells clustered around a single stomata found on the surface of the plant. And you can see that bacteria tend to have much less ability to bypass plant defense mechanisms and penetrate the plant, in contrast to fungi. At the bottom left of screen, we can see how bacteria grow as small slimy cultures, otherwise known as colonies, on nutrient agar. At the bottom of screen, roughly in the middle, you can see a schematic depicting the general structure of a single bacterial cell. And you can see here the general structures of bacteria, including the outer layer called the cell wall. Just to the inside of that, we have the plasma membrane. In this particular example, there's an additional outer layer called the outer capsule. Still to the outside of the cell, you can see a single long flagellum, which looks much like a tail, and this flagellum is used for motion. There's also several smaller hair-like structures called pili. And then to the inside of the bacterial cell, we can see the DNA coloured blue. And of course, the DNA is not surrounded by a nuclear membrane. And this is the major feature that distinguishes prokaryotic cells, such as bacteria, from eukaryotic cells. Also within the cytosol of this cell, you can see many small ribosomes. But of course, bacterial cells being prokaryotes, they lack any membrane enclosed organelles such as mitochondria and Golgi and so on. To the right of screen, you can see what are called gram-negative bacteria. And each bacterium here has a rod shape, and these rods are often connected together. They remain joined together. Okay, now we're going to look at some of the important genera of plant pathogenic bacteria, starting with Agrobacterium. Agrobacterium tumefaciens is the cause of crown gall of stone fruit, although it can infect many other dicots. Agrobacterium vitis causes crown gall of grapevines, and Agrobacterium tumefaciens is quite unique as a pathogen because it has the ability to insert a piece of its own DNA called the transfer DNA, which is found on its plasmid, into the genome of the infected plant host cells, and this causes them to proliferate, thereby forming those galls that you can see in this photo. The transfer DNA also forces the plant cell to produce a unique type of sugar amino acid. The plant itself can't actually use this amino acid, but it provides an important nutrient for the bacterium itself. Taxonomically, Agrobacterium is related to Rhizobium. This is the genus of nitrogen-fixing bacteria, except that Agrobacterium does not fix atmospheric nitrogen. Our next genus is Erwinia, and we have a couple of examples here. For example, Erwinia carotivora, which causes soft rot of vegetables, and also Erwinia amylovora, which causes fire blight of apples and pears. And this particular species is a major quarantine concern because it's present in New Zealand but hasn't yet become established in Australia. In the photo to the left, we have fire blight on apple, and that can be seen as this milky ooze which contains the actual fire blight bacteria, and it's exuding from the infected fruit. Photo in the middle, we have fire blight on apple, and we can see that characteristic crook at the shoot tip, and that's indicated with that white arrow. To the right, you can see an infection by Erwinia carotivora. Our next genus of plant pathogenic bacteria is Ralstonia, formerly known as Pseudomonas. Our first example is Ralstonia solanacearum, which causes bacterial wilt of tomato, pictured here, potato, and tobacco. One particular race or strain of Ralstonia causes bacterial wilt of banana in Central America, otherwise known as moco disease. And a related strain is now causing a serious disease called blood disease of banana in Indonesia. Our next example is Ralstonia syringae, which causes bacterial blight of peas and beans. This is a seed-borne bacterial disease, and it was seen destroying whole paddocks of peas at Horsham in 2003. And here we can see moco disease of banana, which actually displays the same symptoms as blood disease in Indonesia. Our next genus is Xanthomonas. For example, Xanthomonas campestris, PV malvacearum. This causes bacterial blight of cotton. We also have here Xanthomonas campestris, PV citri, which causes citrus canker. This particular disease is common in Papua New Guinea, but it's not yet established in Australia. Let's look at that abbreviation PV that you can see following both species names here. PV stands for pathovar, It describes a bacterial strain or a set of strains that have very similar characteristics. And it differentiates specific strains or pathivars of the same species on the basis of distinctive pathogenicity to one or more plant hosts. Next, we have Xylella fastidiosa. This is a gram-negative rod-shaped bacterium which infects the xylem of the plant, hence the name Xylella. It's transmitted by xylem-feeding insects such as sharpshooters, leafhoppers, and spittlebugs. It was named fastidiosa because it's very difficult to culture in the lab, and so it remained under-researched for many years. Xylella fastidiosa causes Pierce's disease of grapevine in California, and this is the main limitation on grape production in southwest and southeast USA. It's not present yet in Australia, and of course this is a great quarantine concern. Our next genus is Liberobacter. And the two examples here, Liberobacter africanum and Liberobacter asiaticum, represent another group of fastidious bacteria. They cause citrus greening, otherwise known as citrus vein phloem degeneration, CVPD, and it's also known as Huang Long Bin. This is the main limiting factor for citrus production in Southeast Asia and China. It's not yet in Australia, and another major quarantine concern. Corynebacterium causes ring spot disease of potato. Now let's look at some plant pathogenic bacteria that are gram-positive bacteria. Our first example is Clavibacter, which causes ratoon stunting in sugarcane. Next we have Raphaebacter toxicus. This causes ryegrass toxicity in sheep, and it's transmitted by a nematode. The nematode Anguina crawls up grasses and attacks the heads. In ryegrass, it carries the bacterium which then infects the heads, and this produces a yellowish slime that is toxic to sheep. And note, this is a different problem than ryegrass staggers, which is caused by the excessive consumption of ryegrass containing the endophytic fungus Acremonium. Acremonium commonly infects ryegrass without doing any damage to the plant itself. In fact, it benefits the grass by making it more resistant to insect pests. And here we have Streptomyces scabii. This is a filamentous bacterium, meaning that it produces these hyphae-like filaments, just like fungi do. And Streptomyces scabii causes common scab of potato. Now let's take a look at phytoplasmas. Phytoplasmas are bacteria, but they're a unique group of bacteria that lack cell walls. Phytoplasmas are pleomorphic, meaning that they don't have a characteristic shape. They tend to change shape. They infect the phloem of the plant. They're transmitted by insects called leafhoppers. And the symptoms that they actually cause in the plant and the way that they're transmitted to the plant are really more like a virus than a bacterial disease. Here we have one example of a disease caused by a phytoplasma, and this is sweet potato little leaf in Papua New Guinea. Phytoplasmas also cause the disease called tomato big bud, shown here. And Australian grapevine yellows is also caused by a phytoplasma, mainly in white wine varieties. Now let's look at the main ways in which bacterial diseases are controlled. Firstly, by exclusion from the country and from the farm. For example, citrus canker, citrus greening, Pierce's disease of grapevines, fire blight of apples and pears. Second, by planting clean vegetative material. For example, potato tubers that are free of scab, sweet potato cuttings that are free of little leaf disease, and banana sets that are free of Ralstonia. Next, sanitation, cleaning, pruning shears and knives using something like hypochlorite solution, which is common bleach. And finally, selection of resistant cultivars. So, for example, resistant cultivars of bananas were selected and used to control MOCO disease. Finally, let's look at a unique way in which crown gall disease is being controlled. Agrobacterium tumefaciens, of course, is a species of bacteria, and it's identical to a non-pathogenic type called Agrobacterium radiobacter, except that A. tumefaciens contains a tumour-inducing plasmid, and remember that this is a small circular piece of DNA which includes the transfer DNA, the tDNA, that is inserted into the plant host chromosome during pathogenesis, so during onset of the disease. Now, a certain non-pathogenic type of agrobacterium produces an antibiotic called agrocin, and this antibiotic specifically kills the pathogenic Agrobacterium tumefaciens. So this has been developed as a biological control agent for the control of crown gall disease. It's sold as no gall, and it was developed at the Waite Agricultural Research Institute in Adelaide. The commercial cultures are made into a suspension into which the seedlings of stone fruits are dipped before transplanting.